Well, there are actually, platelets are a very important component for blood clotting, but they also derive from a cell that grows in the bone marrow. And that cell that grows in the bone marrow has a stem cell itself. Remember, stem cells are normal. They're normal for all our normal tissues. It's the abnormal stem cell, that's a cancer stem cell, that we're really talking about, and understanding those differences is really very important. One of the key characteristics, indeed, of the cells that give rise to platelets is they have many nuclei. They just don't have one nucleus. They have many nuclei. And recently, Joan Brugge has discovered a mechanism, her laboratory, how cells can accumulate more than one nucleus, and this may turn out to be a very important clue. In fact, we were just talking about this waiting to come on today. Joan, do you want to just mention that area of research?
Well, just because there was a second part of the question.
Well, let's answer this. Let's do this, and then you still
All
have the floor.
I don't.
All right. Talk about an etiology for multinucleated cells.
Well, one of the
seconds.
serendipitous kind of findings that came from research in our lab, which was actually done by Mike Overholzer, who just took a assistant professorship in the cell biology department at Sloan Kettering, was that cells can actually invade into other cells. So when cells are outside of their normal context, which is what happens to tumor cells, when they invade into the microenvironment outside of their normal niches, or when they go into fluid spaces, like in the pleural fluid or ascites. And under these conditions, the cells actually, one cell can invade into the other and sit inside a space within that cell for long periods of time. And under those conditions, if the outer cell nucleus, the host cell to this invading cell, tries to divide, there's often the cell is able to undergo a normal cell division because it has this asteroid size mass inside of it. So the cleavage furrow basically doesn't function. And then you end up with two nuclei inside of one cell. And this leads to enormous genomic instability, and studies in other labs have shown that when a cell doesn't divide normally, such that you have two nuclei within one cell, this leads to improper segregation of chromosomes, so that you have more copies or less copies of individual chromosomes, and this can lead to events that cause some of the alterations that occur during cancer. So this is a kind of serendipitous finding that often happens to all of us in all of our labs when we're studying what we think is one process. Something unusual happens, and if you have a brave and bold enough, usually young investigator in your lab that can follow through on it, interesting things happen.
Or you can be old, brave, and bold, I assure you.